So, um, in this talk, I'm gonna talk about the phenomena of adaptation or homeostasis in general. So, the phenomena of adaptation in biological system are universal phenomena, so that we can observe those things a lot here and there. We maintain our body temperature or body water percentage despite the changing environment, like the temperature of outside or surround. And also our glucose level is also remaining nearly constant, although we have lunch and dinner every day. So these are examples of phenomena of adaptation. And this phenomena of adaptation, or in this case, robust response, this can be identified from those response curves. So when we change the level of certain input chemical species, then we can first put this dose in a lowest level, like 1 millimole, and then the next higher level, second 2 millimole, 3 millimole, and so on. Then every time we put this dose, we can measure the response chemical species concentration at the steady state, and we can respectively like draw the response curve or those response curve like this. And in practice, as you can see here, we will see many dots from the left to the right. And then whenever those dots are concentrated in a nearly horizontal line, then we can say the responses are robust with respect to the given input species. So in this case, the response is roughly the same, independent of the dose, and this is how we experimentally or practically identify the robustness of the response, as you can see in the dose response curve over there. And this is the example, the famous example from the notion of absolute concentration robustness. So when you say a certain thing is robust, we always have to specify what is robust species and also what kind of dose or what kind of parameter that we perturb to see the robustness of the species of interest. So in this case, absolute concentration robustness, ACR is the robustness with respect to all initial conditions. So this is a typical example. So we have two reactions from x plus y to 2y and y to x. And we can write down the corresponding set of ODEs with two lines of equation. And in this case, because there is no pure production or pure decay of x or y species, the summation of those two species is always a constant. So this is the only one conservation law that dynamical system has. And we can write down the algebraic equation that represent the steady state concentration. Then the steady state concentration is given by this formula. So this if our conserved quantity, the conservation law T or summation of x and y is larger than or equal to this threshold, beta divided by alpha, then the steady state is given by this formula. Otherwise, the steady state becomes T comma 0. So basically, this second type of steady state means that the first species X observe everything from X and Y. So based on this steady state concentration formulation, we can draw a phase plane. So you can see that these orange lines, they are these two lines form the steady state set of steady state on the xy plane or non-negative orthant here. So if you start from this initial condition, then it approaches to this vertical line. And if you start this gray region, then it approaches to the orange line, but it touches the boundary on the x-axis. So this is a famous example from the paper from 2010. And our goal in this work is to expand the notion of this ACR to an equally important and practical version. So in practice, as I mentioned, the points or the data that we've observed from the experiments cannot be literally mathematically horizontal. So there should be some perturbations here and there. And the first question we can make here is that, is this approximate robustness just a consequence of adding the perfect flat line and experimental noise? It's a fair guess, but what if the data points or noisy observation from the green asymptote, not the red line? So imagine whenever we do experiment, there will be noises, right? But if we add noise, then we can't be sure if orange experimental data points were actually from the red curve or green curve because they are practically indistinguishable as long as we add additional noise from these two smooth curves. So we wanna shift our focus on this type of those response curve from the perfect flat red line to the more flexible and less restrictive, this green line kind of asymptotes. So here is what we call asymptotic ACR. And this example was from the conventional notion of ACR. So we have the perfect flat line in the dose response curve. So I showed this phase plane in the previous slide, and then I'm going to explain how we convert this phase plane to the dose response curve. So because we have one conserved quantity and the steady state is given by a function of conserved quantity and parameters. But in this case, rate constants, parameters alpha and beta are fixed. So I'm going to draw the dose response curve as a function of the conserved quantity. So we have like three different levels of conserved quantity, 3, 5, 7, for example. Then whenever we start on the initial points on this t equals 3 conserved quantity line, then on this compatibility class, the steady state will be this green triangle. So our conserved quantity level is 3, and then the steady state level is exactly 3, the same. So this is the points that we can get. And then when we increase the level of conserved quantity, then we get this uh, green square, and then this green square will place on this curve, this corner. And then when we increase the level of steady state, level of conserved quantity, then now it moves vertically. So although we increase the level of conserved quantity, now the steady state of x does not increase. So it remains perfectly the same. So that's why we have the stiff corner there, and then the level of x steady state will remain constant. That's how we get this flat dose response curve from the phase plane. And when you add this reversible reaction from x to y, this modified reaction network has the same conservation law because there is no still pure degradation or pure production of X and Y. But now you can see that we add a slight more intensity from X to Y, like this direction, from the right bottom to the left top. So the set of steady state is kind of little elevated from the perfect orange line to this smooth curve. And then if we do the same thing to draw the respective those response curve, then we get this smooth orange curve from the origin to the horizontal line. But now you can see that in the first picture with the conventional ACR, we have the stiff corner at the point beta over alpha, but in this case, we have no such a perfect, stiff corner, then we just smoothly approaches to the level beta over alpha. So this orange curve never touch the horizontal line beta over alpha, but it eventually converges to the level beta over alpha. So you can see that, imagine if we add some noise and then we measure discrete time points with less resolution than these theoretically drawn curves, then we will end up with practically not distinguishable. And also, some someone might think that this second version is not independent of the total concentration because there is initial increment. But the same goes for the conventional notion of ACR, because in order to achieve the perfect independence with respect to all initial condition, we actually had to have like enough amount of X and Y, even for the conventional notion of ACR. So we have to increase the total concentration to escape the initial increment in the conventional ACR example as well. And this is not I would not say this is a limitation, but naturally the original ACR criterion introduced in the science paper, that the network has deficiency 1 and then certain species only differ, certain non-terminal species only differ by that ACR species. Those criteria cannot capture this as asymptotic ACR network, because this asymptotic notion of ACR doesn't fit the definition of ACR in the paper, because this is never like perfectly horizontal flat line. And so if you use the previous criteria, then you would miss this practically indistinguishable notion of asymptotic thing. And this is not only from those exemplary networks, but also there are many asymptotic ACR phenomena showing up in biologically relevant example. So this is again an example from the same paper. And when we computational numerically test several dose response curve from the system, then we can draw the dose response curve of y sub p species like this. And because this example is the example for conventional ACR, the ACR species is y sub p. So this y sub p species steady state level is supposed to be independent of all initial values of the species, like 7 species in this network. And actually it is. So, and because this system has two conserved quantity, X species related conserved quantity and Y species related conserved quantity, when we change the level of X0 and Y0, these two set up basically represent every possible changes in the conserved quantity. So, and you can see this perfect horizontal line whenever we have enough number of X and Y at a certain point. So this is why the Y sub P species is an ACR species. But if we shift our focus onto different species like XPY, this is not an ACR species. This does not satisfy the condition, and actually it does not show the perfect like conventional ACR. But still, when we draw the steady state level of this XPY species along with varying X0 and Y0 value, they show asymptotic ACR. So you see that the steady state response curve, or those response curve, are almost very similar, but the corner over there, they are a little bit smooth. They are not steep corner. So these two curves are approaching to the perfect horizontal line. So this, as I mentioned, this system has these two conservation laws. So although I did not draw those response curve for all 7 species, these two curves represent the variation of all initial conditions. And if we modify this network a little bit by adding one reversible reaction from X sub p to xt, then the species Y sub p no longer satisfy the ACR criterion, and then actually the ACR behavior changes and disappeared. So, but still you can see that although the perfect ACR behavior changes, the steady state response curve along with changing value y of 0 still shows asymptotic ACR. So this corner is kind of smooth corner. And then asymptotic ACR behavior of this XPY species still remains. And if we do the same thing for any possible combination between the initial value change and then respective steady state concentration, then there are many asymptotic ACR species show up here and there. And then, but one thing I have to stress here is the notion of conventional ACR is robustness with respect to all initial condition. So by nature is more restrictive. So you can see that in the first example, the YP species has ACR, and the whole cells in this column, they are like delivered as a whole. So this is just a single ACR because ACR definition of ACR itself is for all species. But asymptotic ACR is with respect to individual input species. So we can test if a certain species has an ACR behavior with respect to every single species. But still, you see kind of the like large block structure, sub block structure on the table, and there's a reasonable and there's a theoretically natural phenomena or natural observation on this table because we can group several species into different conservation law. So for example, among those 7 species, for 3 only contributes the total concentration of X, and the second 2 species, they contribute only to the Y total concentration, and the last 2 species, they contribute both X and Y total concentrations. So whenever we change X or XT or X of P, we essentially change one thing, the total value of X-related concentration. And when we change Y or YP, changing Y and YP, essentially their role is the same. So, and the same goes for the third block. And that's why we have this block structure on the table of this dose response curve characteristics. And these two are other biological example networks, and they do not have a core ACR motif. So because the previous example has a ACR motif in the network, one might think that asymptotic ACR is a mere variant of the conventional ACR, but these two examples illustrate that that is not necessarily the case because these two networks do not have such a core ACR motif, but still there are many species combination. They show asymptotic ACR behavior, as you can see on the table on the right hand side. And what we could prove was that this ubiquity or this pervasive asymptotic ACR is mathematically guaranteed by the network structure. So roughly speaking, if a reaction network has a positive conservation law, then there should be many asymptotic ACRs showing up. So I'm not going to prove it, but I'm going to give you a sketch of the proof with a simple example. So this is the example that we used for the previous graph. And then there were 2 conservation laws, X related and Y related. And among these 7 species, the first 5 species, they contribute the X total concentration. So I want to look at the steady state level of one of the species, let's say the first one, X, in terms of the function of T sub X and T sub Y when we change the initial value or the total concentration of Y. So the point here is that this Y species does not contribute to the level of total concentration for X. So we have this X-related species, and then I pick the one in the outside. So I increase the level of Y and then survey the steady state level of X as Y goes to infinity. So in that case, so this is just an imaginary graph. So when we increase the level of Y0, then at a certain level, we just get the steady state level of X. And then we increase the Y0 value and then again get the steady state level of X. And then we increase the Y0 value and then get the steady state level of X and so on. So we want to look at this purple colored steady state response curve or those response curve for the first species X. And this curve must have three properties. The first one is the steady state curve is always bounded by the total concentration of X because when we increase the level of Y cannot increase the total concentration of X. So T sub X is a fixed constant. And because this T sub X is summation of all the five species and they are non-negative, so this X steady state level is always bounded by the total concentration of X. So you see that the upper bound, this orange dotted line, bounds this purple curve. And then the second one is little intuitive but non-trivial. So this is something that we have to prove actually. So the steady state level of X, so this purple curve cannot oscillate infinitely many times. So roughly speaking, if it oscillates infinitely many times like this, then we can find the zero derivative infinitely many times, and that gives a contradiction to the fact that the steady state level of X is can be formulated by the combination of algebraic function, but those algebraic function cannot have infinitely many zeros. So both facts are contradictive. So that's why this steady state level of X curve cannot oscillate and must converge to the level less than or equal to T sub X. So the second fact can be proven by using some semi-algebraic tools, what we call. And yeah, that's kind of the technical part. And the third one is another simple fact. So the question is, can the steady state value goes to zero because we don't want to see the boundary steady state or zero steady state. So we want to see positive, strictly positive steady state. For the single species X, yes, it can go to zero. But the thing is, when we look at the total five species which contribute to the X-related total concentration, they cannot simultaneously go to zero because their summation must be always T sub X. So if they all go to zero simultaneously, then their summation cannot be T sub X, but it would it would have been like zero, but that is not the case. So at least one of them must be strictly positive. So for that species, we can say that they must show asymptotic ACR because the steady state level converges to the strictly positive and finite value less than or equal to T sub X. And that's why that explains the ACR phenomena shows up in the that shows up in the table over there. So because when we change the level of xt or xp, then they contribute only to T sub X. So then when we look at the steady state level of Y-related species over there, so they are all bounded by T sub Y, and then we can be sure that at least one of them shows ACR, and then in fact numerically, two of them show up asymptotic ACR behavior. And then the same argument can explain this block and that block and so on. And this paper is now under like process of publication. So we submitted and then resubmitted again and again and again. And one fair comment that we recently got is that is this a just a consequence of saturation? Because the argument that I used to prove that there must be many asymptotic ACR is that certain thing is bounded by a positive value and then it converges to the value in between. And to some extent, yes and no at the same time for the question like this, because when we look at some other blocks like this, the notion of asymptotic ACR itself can show up in different condition. So for example, when I change the level, when I increase the level of X or XT or XP, then in principle, the steady state level of X, there is no guarantee to be finite or converges to a non-strictly positive finite value, but still it shows asymptotic ACR behavior. So if asymptotic ACR phenomena was a consequence of saturation, then there should be no such a asymptotic ACR behavior. But still, even if we put more and more and more initial value for X species, the theoretically unbounded species X or XT, they can still show asymptotic ACR. And the same goes for the another block like this. And then so I just explained that those many asymptotic ACR on the table can be explained by the conservation law and the network structure. And in fact, we have a process of algebraic computation that can actually specify every single entry in this table. So when we survey or when we examine the steady state response of XPY species with respect to change of total concentration of X, what we actually have to do is solving the system of ODEs in their steady state condition. So we set everything in the left-hand side 0, and then we get the steady state formula. So we can simplify the algebraic equation in the original system of ODE, and then we get some relationship between the species and then conserved quantities. And the question is, do we know the steady state level of XPY in terms of function of T sub X? Because T sub X is something that we increase right now. And then if we know this function, then we can just let T sub X go to infinity and then see what the steady state value of XPY is. So we just started from the steady state formula, and then we had to do some multiple steps of algebraic computation. So they are all about like function simplification and then elimination. So we can simply write down T sub X and T sub Y as a function of several species. And then these are all combination of different rate constants. So we just redefine simpler parameter A, B, C, and D. And then finally, we get functions of x, p, y, and y for t sub x and t sub y. And we further simplify it. And finally, what we can get here is we get like ty as a function of t sub y and so on. And then we just replug this value in the first equation. Then we get a quadratic equation with respect to xpy steady state, and then several coefficients are given by t sub y and t sub x and a, b, c, and d. So they are all parameters. Because this is a quadratic equation, we can actually apply the root formula, but that is not generally applicable. What if we have like equations, polynomial equation more than degree 5 or something, then we don't have such a root formula. So instead of solving this one explicitly, we want to do indirect way. So we have this implicit function. So we just define this implicit function as the capital P, and then we deduct the fact that this implicit function has the highest order term of lambda. So in this case, lambda is xpy. So, oh, sorry. So in this case, lambda is something that we let go to infinity. So t sub x is lambda, and lowercase x is the steady state xpy. And because this algebraic equation is always zero, so what we can say here is that when we let lambda go to infinity, because this is the leading term, the highest order term, q sub x must be zero to compensate this increasing dominant term, lambda to the power of m, and that gives this q sub x, this value xp steady state of xpy must be a root of q sub x, q of x. That's how we make this capital P function always zero. And that's why when t sub x goes to infinity, this steady state value xp sub y, x sub p of y, is given by this explicit function. So what we did here, instead of driving this function as a function of t sub x for all value, we just examine the value of this function when the input argument t sub x goes to infinity. So, and this is generally applicable as long as we have this implicit function. So that's how we can show that xpy admits asymptotic ACR with respect to x. And the same argument can go for any entry on the table. So here's this quick summary. So asymptotic ACR is ubiquitous, and then reaction networks with positive conservation law have eventually asymptotic ACR here and there. And then this is not just a perturbation or little small variants of the conventional ACR. That's it. And this is my end of my talk. And thank you for listening.